Hello everyone, welcome back to another Shell IB Chemistry video. Today we are doing the last part of covalent bonding, which involves chromatography. So what is chromatography? Chromatography is a separation technique which enables the separation of mixtures. There are two main types of chromatography, which include paper chromatography, which is the most commonly taught up to this point, but we also have something called thin layer or TLC, which is thin layer chromatography. How chromatography works is that it makes use of the principle that components in a mixture, when they are dissolved in a fluid or mobile phase, that they will flow through another material in a stationary phase or a solid at varying rates due to different intermolecular forces mixture between the different components. The rate or the distance which is traveled by a mixture will vary dependent on these intermolecular forces present. Let's get into paper chromatography and use this as an example to better understand how this works. So in paper chromatography, you'll typically have a beaker with possibly a stirring rod or a pencil, which will hold up a sheet of paper. This paper will have a line, a dotted start line in pencil, not in ink, or else this will move up the paper along with whichever mixture or dye sample you wish to put on it, tampering with your results. And you will have your solvent up here just touching the paper but below your start line. Always below your start line. So this solvent over here will travel up the paper by capillary action. Now, if we think of capillaries, this probably reminds you of the tiny, tiny little blood vessels that are present in the body that you may have heard of before in biology. Now, that's exactly what this is. So, capillary action is the upwards movement of a liquid through a very slim, very small tube. Now, this is made possible because of the intermolecular forces present in your mixture, affecting its solubility. Paper contains cellulose fibers which have a hydroxyl group, an OH. This means that this solid is able to have intermolecular forces and interactions with mixtures because it can have hydrogen bonding, as we can see by this hydroxyl group over here. If we were to have, say, water as our solvent, we have our slightly more positive end here and more negative one down there. Now, as we can see, this molecule has an overall dipole, meaning that it is not a neutral molecule, and as it is also able to form hydrogen bonds with other oxygens, nitrogens, or fluorines. Remember, these are elements within the electronegative triad. These will be very attracted to each other, meaning that there will be a lot of adhesion and cohesion. Cohesion refers to the attractions amongst molecules, keeping them together. So water is a very cohesive substance because it's able to have these hydrogen bonds. And adhesion refers to water's ability to adhere to another substance, in this case to paper, because of this hydroxyl group, permitting, as we've mentioned earlier, hydrogen bonding. Now, substances in the mixture which can form hydrogen bonds with this OH group will be more attracted to the stationary phase, or the paper, than those which form weak intermolecular forces with it. Now, if you have weaker intermolecular forces, this means that you will get less adhesion to the stationary phase, so this will not travel as far on your paper chromatogram than the opposite would. Next up, let's look at TLC, or thin layer chromatography. Now, thin layer chromatography looks quite similar to paper chromatography, and it works in a similar way as well, but the main difference is that TLC has a different stationary phase. So the stationary phase, or what is in a sense absorbing your mixture in TLC, will be a very thin layer of an inert substance such as silica or alumina. This will be placed or supported on a flat, unreactive surface such as glass, and your mobile phase or mixture is your solvent, and silica and alumina both contain hydroxyl groups to form the hydrogen bonds with components in the sample, and these components in the sample are absorbed onto the surface of the stationary phase. And just as in paper chromatography, depending on the strength of the intermolecular forces, the separated components will travel specific distances throughout the plate. Last up, let's look at how to calculate an RF value and what exactly this is. So RF value stands for retardation factor, and this demonstrates the extent of separation of the components molecules in the sample, which is dependent on their solubility in the mobile phase and the extent of the adhesion to the stationary phase. So RF value is used to quantify the distance a component travels relative to its solvent. So you'll have your distance traveled by the component over the distance traveled by the solvent. And these are helpful values to calculate because they can be used alongside other types of analytical data to deduce the compositions of mixtures. That is all for today. Hope you enjoyed and that this was in some way helpful.